Hello dear students, welcome to Biochemistry by Revati. I am Revati and in today's class we will learn what is stereochemistry in carbohydrates, right? You may wonder what do this right, left and mirror images have to do with sugars, right? Let's find out. What is stereochemistry? This we have already studied in the previous class, right? Let's recall quickly. What is stereochemistry? It is the study of 3D arrangement of atoms in a molecule, right? What is stereochemistry? It is a 3D arrangement of atoms in a molecule. Now we deal this concept especially in carbohydrates, right? So here this stereochemistry helps us to understand how the molecules like glucose can have the same formula but different structures and properties, right? Why it is? Simply because the arrangement of atoms are different in a space. Is it clear? Now let's see why stereochemistry is important in carbohydrates, right? So here carbohydrates like glucose and galactose are stereoisomers. What are stereoisomers? Stereoisomers are the compounds that have same molecular formula but different structures and properties. Means they have different spatial arrangement of atoms, right? Now that difference will make the enzymes to recognize to a particular substrate only, right? So here, what galactose and glucose. So they are stereoisomers, is having the same molecular formula but different spatial arrangement of atoms. Now this difference makes enzymes to help and recognize the specific substrate. Now if you see, for example, glucose, our body can easily digest D-glucose but not L form. Why? Because the enzyme means the enzyme hexokinase, it is stereospecific, means it acts particularly on D forms but not L forms, right? So this is because the chirality of the enzyme which have been evolved to fit only to the specific D forms, like a lock and key, means a particular lock fit to a particular key only, right? Is it clear? Now let's break down the important terms you must know, right? First, chiral carbon. In the previous class, we have studied that chiral carbon is the one that is attached to the 4 different groups, right? Now we are taking example in carbohydrates, that is glucose. If you see the Fischer projection, means the open chain projection of glucose. Glucose is a 6 carbon compound, means it contains 6 carbons, right? So if you see the structure, start numbering: carbon 1, 2, 3, 4, 5, 6. So it is 6 carbon compound, right? So now here we are saying that glucose contains chiral carbons. Let's see how. So here Now I am saying that this glucose has 4 chiral carbons. Carbon 2, carbon 3, carbon 4 and carbon 5. Let's see how. If you see the second carbon, it has been attached to 4 different functional groups. Aldehyde group, hydrogen, hydroxyl, carbon 3. If you see the third carbon, again it is attached to 4 different carbon 2, hydroxyl, hydrogen, carbon 4. Again fourth carbon it is attached to, like that, vice versa. So here now if a question has been arised, glucose contains how many chiral carbons, means you have to answer it as 4 chiral carbons. Glucose contains 4 chiral carbons. Is it clear? Second term is enantiomers. What are enantiomers? This we have already studied in the previous class. These are mirror images. Means mirror images or mirror image stereoisomers. Means they cannot be superimposed on each other. Means they cannot be placed on top of each other. Like example, if you take left hand and right hand. Left hand will not fit onto the right hand, right? Or left hand glove will not fit onto the right hand, right? So here we are taking in carbohydrates example as D and L forms of glucose. Let's see. So here glucose is a how many numbered carbon? 6 carbon compound, right? Now let's see what makes the difference between D form and L form. Now third one is epimers, right? Epimers are the diastereomers, means they have same molecular formula but have different bonds, means the arrangement of atoms in a molecule is different, means they have different physical properties and chemical properties also, right? Because they have different structures, only they have same molecular formula. Example, we are taking in carbohydrates again, example glucose and galactose. So glucose, both are 6 carbon compound only, but there will be a difference in the arrangement of atoms. Start numbering 4, 5, 6, right? So here for glucose only only on the third carbon atom will be a change in the difference of atoms. So this is the glucose. Now let's see galactose. One, 2, 3, 4, 5, 6. CH2OH. So there will be a change in the difference in fourth carbon configuration. If you see epimers will differ in fourth carbon configuration, right? You see the both structures, there will be the difference in the arrangement of atoms in the fourth carbon arrangement. The hydroxyl group is on the right, here hydroxyl group is on the left. So this is the difference between the glucose and galactose. Is it clear about epimers? The final term, that is anomers, right? So here now these anomers are formed during ring closure. Let's see what is actually this. Example, alpha and beta glucose forms. So if you see the structure of glucose, open chain means we call it as what? Fischer projection. Fine. So it is the Fischer projection. About this you study in further classes. This is the Fischer projection of glucose. Now let's see how this glucose will turn into the ring structure. Let's see in the ring form. If you see, how many carbon atoms? 1, 2, 3, 4, 5, 6, right? So here, start numbering in the Fischer projection 1, 2, 3, 4, 5, 6. Now taking the first carbon. First carbon has what? It has hydrogen and oxygen, double bonded, right? This is the first carbon. Second carbon, H, OH. Third carbon, top and bottom we have to write. Here it is left and right. Here it is top and bottom. So here OH, H. Fourth carbon, H, OH. Fifth carbon, fifth carbon, H, H, this OH. Now we'll see what will happen. To the fifth carbon only, sixth carbon atom will be attached. So it is sixth carbon atom. Have you understand? So here this Fischer projection we are making into the ring, means Haworth projection. Let's see, 6 carbon atom into a ring form. This is the first carbon which is having the aldehyde group C double bond H, and second carbon HOH, H on the top, OH on the bottom. Now this OHH on the top, bottom, fourth carbon atom. To the fifth carbon atom, hydroxyl group we are writing on the side, and to the fifth carbon atom only this CH2OH is attached, right? Sixth carbon atom is attached to the fifth carbon atom. Now let's see what will happen. Now to this, how the ring closure takes place, let's see. So first carbon, second H, OH, third OH, H, fourth H, OH. This is the fifth carbon atom, and to the fifth carbon atom only CH2H is attached. This is the sixth carbon. So here we are having hydroxyl groups, right? Now this here the proton transfer will take place. Proton transfer means the hydrogen atom will bind to the oxygen, right? So let's see what will happen. Now there is a transfer of proton. Now this H will combine with oxygen to form hydroxyl. Is it clear now? This is the alpha D glucose. If the H is on the top and OH is on the bottom, then it is alpha D glucose. We call this projection as Haworth projection, means ring closure. Now let's see to the beta form. There will be only a difference in the configuration of HOH only. Let's see. So here H on the top, OH on the bottom, right? If it is on the top and H on the bottom means it is beta D glucose. Is it clear alpha and beta D forms? To clearly understand the concept of stereochemistry in carbohydrates, now let's see with a real life example, right? So, have you ever wondered why some people can't digest milk? The reason lies in a sugar called what? Lactose. And an enzyme? Lactase. So, here what is actually a lactose? It is a disaccharide. Disaccharide means it contains two sugars. What are those? Glucose and galactose. Now these two sugars are bonded by which bond? Beta 1,4 glycosidic bond. So here the enzyme lactase, it is stereospecific. Stereospecific means it will break when the beta 1,4 glycosidic bond is present between the glucose and galactose, right? So here if the hydroxyl group, if the orientation of the hydroxyl group on the galactose is correct, then only it will break down. Otherwise it will not break down because it is the stereochemistry of the beta bond, right? So here, now what will happen in the lactose intolerance people? Let's see. If the lactase enzyme is inactive or non-functional, means it is, if it is not functioning, what will happen? The lactase will it break? It will not break. What it will not break? It will not break the beta 1,4 glycosidic bond present between the glucose and galactose. If the bond is not broken, means what will happen? The lactose isn't digested. Without digestion, it will move to large intestine, which causes what? bloating, diarrhea, or sometimes or and discomfort to the body. So it's not just the sugars that matters, but it's the 3D shape and the enzyme ability to read the shape. So that's the magic of stereochemistry in carbohydrates. Have you understood? That's all for today's video. I hope you have understood the concept clearly. If you have any doubts regarding the concept, please comment on your doubts, right? And if you found this video helpful, please like, share and subscribe to Biochemistry by Revati for more simple and clear explanations. Please do support me, and in the next video we will learn the concept of stereochemistry in amino acids, right? So until then, keep learning and stay curious. See you in the next class.